Welcome everyone to Scientists Warning Europe's Nature Day conference. The first Alliance of World Scientists Warning in 1992 was endorsed by 1,700 scientists, including more than half of Nobel laureates at that time. In 2017, a second warning was raised that attracted over 20,000 scientists' signatures. And in 2019, a climate emergency was declared, supported by over 15,000 scientists. On nature, the World Scientists Warning says we must protect and restore Earth's ecosystems that contribute greatly to sequestration of atmospheric CO2. They go on to say more, but without further ado, I'll now hand over to Bill Dowling, who will introduce our next speaker.
Thank you, Brian. It gives me great pleasure to introduce Dave Wilkinson, who has very wide interest in ecology and the environmental sciences. He's taught at Liverpool John Moores University for almost 25 years and is now visiting professor in ecology at the University of Lincoln and an honorary research fellow in archaeology at Nottingham University. His book, Fundamental Processes in Ecology, an Earth System Approach, won the British Ecological Societies Book Award for 2007, and he is currently writing a book on ecology and natural history for the Harper Collins long-running New Naturalist series. His talk today is entitled Microbes, Ecology and Global Change. He points out that microbes are often overlooked, but crucial to most ecological processes. And they're pretty small things, aren't they? I discuss the effects of human-driven global change on microbes, especially in respect to soil microbes and their job to do in agriculture. Over to you, Dave.
Okay, thank you very much. Let me see if we can now lost the... We had the PowerPoint, but
hey Dave, if you just stop your screen share for 1 second, and then when you go to restart it, it should give you an option to click on the PowerPoint option. So it might be worth having your PowerPoint focused first. Yeah, that's not worked. If you just focus on...
there we go, it's come back now. That is back where we were last time.
There we go.
Perfect, thank you.
Go ahead, Dave.
Okay, so hopefully that is now working. Worked in practice, and of course went in itself for the real event. Okay, so what I want to do over the next hour is to say something about microbes in the context of global change. And clearly this is an enormous topic, so I'm going to have to sort of focus down on a few things. So really this talk falls into 3 parts, and then hopefully there's some time for some questions at the end. And the first part, what I want to do is to sort of give a general introduction to microbes, because by definition they are too small to see and therefore tend to be very unfamiliar to people, and then say something, particularly using late microbes, about interactions with climate change and microbial ecology, and then focus the final part of the talk really on soil microbes, particularly in the context of agriculture, because clearly agricultural production is a major thing that where we are changing the planet and where we are interacting with climate change and many other changes. So the pretty pictures that you see on the slide here are actually produced by Joe Leidy in the 19th century, at a time when you couldn't take photos down microscopes, so it helped to be a really good artist at the same time. And these are here, 1, because they're pretty, but 2, because they're actually illustrations of testate amoebae, shell-forming amoebae that live in both in soils and water, and they're one of the groups of microbes that I particularly study. So if you think about biodiversity normally, you think about this sort of thing. So here we've got a collection of bits of biodiversity, some from Britain, some from South Africa. And these are the sorts of things that you would see on a TV natural history program. And there's no real microbes here at all. We've got plants, we've got animals, and we've got some fungi and lichens. So the nearest thing we have here to microbes are the fungi and the lichens. Many types of fungi are entirely microscopic, and in general anyway, the fungi and lichens, if you pick up a big textbook, university level textbook on microbial biology, you will find that there's usually a chapter on fungi. They sort of get themselves in there, even the bigger ones, as sort of honorary microbes. But most of what you sort of see when you watch a program on the telly on natural history or on conservation or threats to biodiversity are all these big things. Now, clearly, when you sort of lecture on this sort of thing quite a lot, you end up sort of drifting into sort of, you know, little set piece sound bites to make a point. And my sound bite for this sort of thing is always, if you imagine all of the organisms that have made David Attenborough's bank account really large, that's actually a tiny proportion of total biodiversity, because as I'll show in a few minutes, most biodiversity on this planet is actually microbial, and microbes hardly ever feature on TV programs on natural history and conservation and declines in biodiversity. So just to introduce a few terms for those who don't know, microbes, in fact all of life, falls into one of two sorts. There's our sort of life, which are eukaryotes and have cells like the bottom one that you see in this diagram, which I've taken from one of the leading microbiology university level texts that's gone through multiple editions. And these are eukaryotic cells. And they have in them a whole collection of things that are called organelles. The idea is that they're rather like the organs that we have in our body. Heart does one thing, lung does another thing, your liver and pancreas do other things. And within a eukaryotic cell, there are a whole collection of these organelles that do different functions. The prokaryotes, which include the bacteria, are different. They have much less structure within their cells, as you can see in this top thing. So prokaryotes are made up of what we call proper bacteria, and also a group called the archaea, which if you're unfamiliar with, until about the 1970s, we'd have called all of the prokaryotes bacteria. But during the course of the 1970s, molecular methods looking at RNA and DNA, the genetic material in cells, it became apparent that actually within bacteria, there were two very different sorts of things to be found. The archaea on one side and the true bacteria on another. So bacteria, archaea, prokaryotes, much less structured and tend to be smaller cells, and the eukaryotes are bigger, but microbes fall into both groups. There are clearly prokaryote microbes. Indeed, you know, most prokaryotes are microbes. They are very small bacteria and things, and only when they clump together into big lumps can you actually see them with the naked eye. And then there are the eukaryotes. We are eukaryotes. All of the things that have made David Attenborough wealthy are eukaryotes, but still most eukaryotic biodiversity is actually made up of microbes. So if we look at a diagram like this, and it looks horribly complicated, but I just want to use it to make a point. There are multiple versions of this sort of diagram out here. I've particularly stolen this one out of this paper by Charlie Weaver and co, because I like the "you are here" in. So all of this stuff up at the top are all prokaryotes. This has been put together using genetic information. Okay, so looking at the genes in organisms and how different they are. And so you can see that most of genetic diversity is actually prokaryotes. All of the things at the top are bacteria, although they fall into two big groups, and maybe at some point we might split them. And then down at the bottom, they're mainly archaea, with this collection of green things with the "you are here" on, which are the eukaryotes. So if you look at things at a genetic level, looking at the genes and looking at the DNA and RNA of organisms, eukaryotes are a very small amount of diversity. Most of biodiversity at a genetic level is made up of by the prokaryotes. So we can think, well, let's think about the eukaryotes. And this is now ignoring all the prokaryotes and just again using modern genetic information to look at the diversity to genetic level of eukaryotes, the things with our sorts of cells. And the key thing to note is that All right, again, it's a complicated looking diagram, but let me show you, let me do the you are here again. Bonk. That little branch there are all of the animals. That's nearly everything that ever appears on a telly program about natural history, biodiversity, conservation, or anything else. A tiny little twig. And the majority of the things you're seeing here, the plants are here is another twig, the fungi is another twig, and some other things that you can see, the brown seaweeds are part of another twig, but nearly all of these twigs are totally microbial. And even the twigs that aren't include lots of things that are too small to see with the naked eye and include many, many microbes. So if you look at things at a genetic level, then nearly all of biodiversity is microbial. That's true overall. And even if you ignore the bacteria and the archaea, even if you ignore all of the prokaryotes, it's still true of the eukaryotes that nearly all diversity of eukaryotes is actually microbial. And if you're looking at this going, what a lot of complicated names, and I don't really know any of these things at all, that would actually be true of most people with a biology degree. Won't know most of these groups. Many of them barely feature even in a biology degree. However, that's looking at things as a genetic level, and at a genetic level, almost all life is made up by microbes. But I'm not that sort of biologist. I get involved with using genetic techniques to look at microbes, but I'm basically an ecologist. And let's think about the history of all of life on Earth. And this is a nice, neat little summary. It actually comes from a book by Maureen O'Malley. She's one of the very few philosophers of science who've ever taken an interest in microbes. And if you have any interest in the intersection of biology and philosophy and what thinking about microbial biology, what sort of interesting philosophical questions it raises, then if you don't know this book, it is one to look at. So what we've got is the time span of life on Earth here. So we start over in the left-hand corner at 4.5 billion years ago, around the time the planet's formed, and all the way through to now. And there's a whole collection of things turning up. The first prokaryotes, the first cyanobacteria, the great oxidation event, which is when photosynthesis starts to allow us to have lots of oxygen in the atmosphere, the first eukaryotic microbes, and then eventually metazoans, animals and things turning up. And the point I want to make is that for most of the history of life on Earth, all of this black box from the earliest life, and there are not clearly arguments about when the first life appears, but about 3.8 billion years ago, 3.8 thousand million years ago looks to be reasonable for some of the first life. From then until only about half a billion years ago, all of life on this planet was microbial. So for the bulk of the history of life on Earth, all ecology was only microbial ecology. And still today, microbes are crucial to ecology. Still today, microbes drive an awful lot of the ecology of the planet, even though if you were to look in a standard ecology textbook, you would actually find relatively few microbes there, and focusing on plants and animals. And yet microbes have for most of the history of life on Earth been the only game in town when it comes to ecology, and still they are absolutely crucial to most of the key ecological processes going on the planet. And just to help illustrate that, this is a fossil. This is about 2.6 billion years old in South Africa, and this is the remains of sheets of photosynthetic bacteria, probably living on the base of sort of shallow water. So this is a thing called the stromatolites, and these are early and very distinctive fossils. You can still find stromatolites in some parts of the world, and you get the cyanobacteria forming these cushions on the bottom of shallow water. The one that if you're got the likes of David Attenborough before he's quite so old and didn't quite always jet around the planet, was actually in a place called Shark Bay in Australia. And the reason you can still see stromatolites there is because it's very, very salty, so salty almost no animals can survive there. And so once animals turn up, they tend to eat these things and destroy them. So what we've got here is a layer of photosynthetic bacteria on the bottom of the sea, and bits of sediment and things get over it, and it grows over the top, and you get this onion-like structure. And so we have these photosynthetic, almost certainly photosynthetic things on the ocean bottom 2.6 billion years ago. So I'm primarily, though, an ecologist, and so rather than thinking about the long-term history or indeed the genetic diversity, clearly what matters a lot in ecological processes is the biomass of an organism. How much is there? You know, an organism can't alter the carbon cycle, for example, very much unless there's a big body of that organism to be interacting in a big way. There's got to be a lot of carbon going through those organisms for them to have a significant effect. And so it's interesting to ask, well, from the point of view of biomass, amount of material and weight of organisms, which dominate. And this is the most recent and up-to-date attempt at doing that by an Israeli team. And as you can see, what we've got here are different groups, and then their mass in gigatons of carbon, just how much carbon is estimated to be in all of the plants on the world, all of the bacteria in the world, and so on. And because that's not an easy thing to do, what the authors sensibly did is to make a guess of their level of certainty. So you can see that for plants, there's a high level of certainty. It's easy to work out how many plants there are on the planet. You can walk around and count them. And nowadays, you can actually, from a satellite, easily quantify the numbers of plants. And we're just getting to, and use a paper in the issue of this week's Nature, where over a large chunk of Africa, they had been actually able to count every individual tree using satellite technology. So plants we know quite well. Bacteria being invisible to the naked eye, much harder to count, and so there's much less certainty. So on their best guess, most of the biomass on the planet is in plants, followed by bacteria, then followed by fungi, and so on and so on. Animals very, very down the bottom there, and nearly all of that biomass in animals is actually in marine invertebrates, not things actually living on land. Clearly, if you look at the uncertainty there, then because bacteria are so uncertain, it's possible there is a weight of bacteria equal to or even in excess of the weight of plants. And indeed, when this paper came out, one of the things that surprised quite a lot of people was the low number they got for bacteria. The best guesses for bacteria before this had actually been higher than plants. So it could well be that bacteria are at least as much carbon in them as all plants on the planet. Certainly they're running second. And the other thing that's interesting and counterintuitive and is extraordinary, and yet most people don't know it, is where most of these bacteria are. Most of the bacteria on the planet are actually underground in cracks and pores in the rock, going deep into the rocks. Now, they're very sparse down there. They're in little cracks and pores in the rocks. There's very little food for them down there, so they live very slowly and at a very low density. But because there is just so much volume of rock under us, under our feet, there are colossal numbers of bacteria in what's often called the deep hot biosphere. And only a few decades ago, when this was originally suggested, actually by a rather interesting theoretical physicist called Tommy Gold, people said this is absolute nonsense. There can't be lots of bacteria down there. We now know that Gold was right. We now know that there are colossal numbers of bacteria and archaea and prokaryotes living deep in the rocks under very hot conditions because they're much nearer the hot Earth core. Other bits of Gold's idea, he also had ideas of what this implications of this for the formation of oil and things haven't stood the test of time. But his basic idea that there's a colossal amount of bacterial life down deep into the Earth rocks turns out actually to be correct. So a lot of biodiversity is actually deep underground. However, plants and bacteria win. So you're asking where within life most carbon is, it's in plants and in bacteria. And then, things like animals, barely any at all, by comparison with what's looked up in plants and what's locked up in the prokaryotes. So to think briefly about photosynthesis, because again, this is something where people have the wrong impression. Photosynthesis is something done by plants. The trees, we get very excited, and quite rightly get very excited about forests and loss of forests in relation to the carbon cycle, in relation to global heating and things. But actually most photosynthesis on this planet is being done by much smaller things. And about half of all the primary productivity, half of all the photosynthesis on Earth is actually being done in the ocean by microbes, rather than on land by big things that we can actually see. And this involves prokaryotes and eukaryotes. And again, really quite important, because when these things die, if their bodies drift down to the bottom of the ocean, they get entombed in the muds at the bottom of the ocean, and that can take carbon out of the system for a very long time. Alternatively, if you're on land, at the moment, I can look out of my door and I can see the leaves coming off the apple tree down the garden. But those leaves, yes, they've got carbon in, but they will be broken down really quite quickly by microbes and things, and that carbon will go back into the atmosphere. So actually, far more carbon is locked up from photosynthetic organisms in the oceans than is locked up from photosynthetic or long-term from photosynthetic organisms on land. And I want to sort of introduce you to a really remarkable little thing. And this remarkable little thing is Prokaryote. And this is what we call our phytoplankton. So it's a phytoplankton, it's photosynthetic plankton, but it's pico because it's absolutely tiny. And there are several things that are really extraordinary about this thing. It was only described in, you can see, in 1988. I'd already got 2 degrees by the time this thing, was first described by science. And all right, I'm going grey, but I'm not absolutely decrepit yet. This wasn't known when I was an undergraduate. And yet, if you were to say, take a tube of this sort of size and dip it into the ocean, anywhere except at high latitude, in a tube just as big as this, you would have about 10 million Prokaryote cells. It is extraordinarily abundant in the oceans, and indeed is possibly the most abundant organism on Earth. Now that is extraordinary. What might be the commonest organism on Earth wasn't even known to science when I was an undergraduate. It illustrates 2 things. It illustrates how little we know about microbial ecology and how much we are now learning with modern techniques. And it also illustrates, these are photosynthetic. They're colossal numbers of them. Yes, they're all very tiny, but because they're in there in such enormous numbers, they are doing huge amounts of photosynthesis. And these photosynthetic prokaryotes, like Prokaryote, are what we call cyanobacteria. Again, if you're old enough, they used to be called often blue-green algae. They're blue-green in colour, algae, but they're actually not eukaryotic organisms. They are bacteria, they are prokaryotes. And many of them can be actually bigger. And what I want to do is to say a little bit about blue-green algae in relation to these cyanobacteria, in relation to global change and global heating, before moving on and talking about microbes in soils, particularly in the context of agriculture. I had a long-term sort of very side interest in cyanobacteria, ever really actually since I was a master's student in the mid-1980s. I attended a number of lectures by Brian Whitten at the University of Durham, who was one of the key figures in the biology of cyanobacteria in this country. In fact, he's still interested deep into retirement in cyanobacteria. And one of the ways in which cyanobacteria interact with ourselves, and I'm going to now talk mainly about freshwater, because most of my work tends to be on, when it's aquatic microbes, on freshwater rather than marine. In fact, actually, my very first marine microbiology paper has only just actually come out. So cyanobacteria have these things that are called algal blooms, and they turn the water, as you can see here, bright sort of bluey green. This is actually a river in subtropical China with a cyanobacterial bloom in progress. And if you look at the bigger cyanobacteria under a microscope, many of them actually form these long chains. This actually isn't a photograph of a Chinese sample. This is actually a sample that I took from Windermere a few years ago. But one thing to notice on them is these bigger cells in the middle. And these are specialist cells where they can actually fix nitrogen. And so one of the reasons that cyanobacteria can be so successful is not only can they photosynthesize, but they can actually take nitrogen from the atmosphere and from the water and turn it into forms which they can use. And very few organisms can do that. So actually getting nitrogen is often actually a problem for organisms. So they are nitrogen fixers in the same way pea plants are, but again, pea plants can't do it. They do it with the help of bacteria in their roots. Cyanobacteria can fix nitrogen. But under certain conditions, you can get these large blooms of cyanobacteria that turn the water a completely different color and are a major issue from the point of view of human use of these waters. Amongst other things, they produce a wide range of toxins that actually can be very, very bad for you. And you will nowadays see signs, in summer sometimes by a lake, do not swim, cyanobacterial bloom, and in particular, do not let your dog swim, cyanobacterial bloom. Dogs, for some reason, seem to be particularly susceptible to these toxins. So I'll show you a little bit of data from work that I've done in China with a team from the Chinese Academy of Sciences Institute for the Urban Environment, because China, well, the two statistics on these slides which show why water and water pollution is just such a big issue for China. China has about a fifth of the global population of humans, and yet it only has 7% of the global amount of fresh water, and that's probably declining over the next few decades because a lot of it comes from the Himalayas, from glaciers, and these glaciers are now melting and retreating, and there is a concern that that 7% is actually going to drop. And so water is in relatively short supply in China, and now a lot of it is distinctly polluted, which again limits what you can do with that water. So water pollution is a big issue in China. So just to illustrate a little bit about cyanobacterial blooms, then let me show you these data from a couple of reservoirs in subtropical southern China. So these are two reservoirs that link one to the other, one immediately above the other. And what I'll do is show you some data from this paper that we published last year in the ISME journal, looking at about a 6-year run of data with repeated sampling from these reservoirs. Sampling starts at the height of a cyanobacterial bloom, then the bloom dies off and we carry on sampling, and that continues until my second visual aid just fell on the floor as I wave my hands about, but it's all right, it will come back later, until into the next cyanobacterial bloom. So this is indeed one of the reservoirs, and collecting a whole collection of data here. The guy on the left there has got a standard sort of old-fashioned plankton net there, a net of very, very fine mesh, so that all but the smallest microbes will be contained within it and go into the collecting tube, and you can drop that down through the water, haul it back up and sample the microbes through the water body. You can see tubes and things at the back of the boat there squirting out water, and there we've got a little pump and generator, and you can drop a hose down to a depth that you want to sample and then pump water up from various samples. Well, the guys in the front here have got a variety of high-tech devices for sampling both the chemistry and the physical aspects of the water, everything, temperature and so on, to aspects of chemistry, oxygen levels and things. So here is a little bit of the data. So we've got these two reservoirs. The top two graphs show you the numbers of cyanobacteria, the amount in a particular volume of water. Note though that it's on a log scale. So if plotted on a non-log scale, the blooms would look much bigger, but you can see there's high numbers to the left. They then drop and then start to come up again towards the end of the sampling. Whereas the two bottom panels there are showing the percentage of all the microbes in our samples that are actually made up from cyanobacteria. And the height of the bloom, there's so many cyanobacteria that basically all, close to 100% of everything that you're seeing down a microscope is actually cyanobacteria. And this has a big effect. So what you're seeing here is water transparency. And you measure this fairly straightforwardly by just lowering on a string a disc into the water and working out at what depth you can no longer see it. And basically at the height of the bloom, only a few tens of centimeters down, you're losing sight of a disc that's about this big. Whereas when the bloom's not there at all, that you can still see it down to nearly 2.5 meters depth. And clearly this has implications for what depth organisms can photosynthesize. So cyanobacterial blooms are completely altering the ecology of this water by altering the amount that light can get down to them. They're having colossal human implications by their effects on water quality, and in particular, the amount of toxins and things they're putting into the water, which then you're going to have to spend quite a lot of effort treating that water to before you can actually use it for anything. And this interacts with big things in global change. So this is the lower of these reservoirs. And there are really two things that drive cyanobacterial blooms. One are nutrient levels. And one of the things that we've been doing to the planet that hasn't had the attention that global heating's had is we have made most systems vastly more nutrient rich. Everything we've been doing with agricultural things and large amounts of fertilizers, and I'm going to come on and talk about agricultural soils and microbes in a minute, has put vastly more nutrients into the system. So large amounts of nutrients get washed into systems like these reservoirs. The other thing that affects cyanobacterial blooms particularly is temperature. The warmer it is, then the faster these things grow and the bigger problems that you've got with cyanobacterial blooms. So here we've got a really nice example, this is what I put into this talk, of two of the really dramatic things that we've been doing to the planet on a global scale, making it much more nutrient rich and making the climate water interacting to make these events, these cyanobacterial blooms, more likely and a much bigger problem. And you can sort of do ballpark calculations. And if, for example, you were to say, well, global warming, that's harder to do things about than, for example, reducing nutrients. If you were trying to reduce cyanobacterial blooms by reducing nutrients, you'd need to reduce them by about 1/3 to counteract just 1 degree C of warming. These are clearly very, very ballpark figures, but it shows the difficulties that you've got and how these two major things that we're doing to the planet are interacting to give us increased problems with cyanobacterial blooms in both freshwater systems and also in particularly shallow marine systems just off the coast, where you're getting large amounts of nutrients being washed in from things that we were doing on the land, coming down the rivers and going into the seas. And we have huge problems with cyanobacterial blooms in shallow marine systems as well. So what I want to do for the rest of the talk is to move on and say something about soils in general, and particularly agricultural soils and their sort of microbes. And I'm going to focus particularly after the first few slides on eukaryotic microbes in soils, because these are the things I particularly study. So the things that used to be called protozoa, we would now call them protists, but particularly the protists that actually actively ingest other organisms and things in the soil, things that we used to call protozoa, and also then a little bit about fungi. Say they often get treated along within microbiology texts, and I'll say something about mycorrhizal fungi towards the end as well. But just to give you an idea how much stuff in the way of microbes that you have in a soil, if you have 1 centimeter cubed of soil, which is roughly what I've got at the bottom of this tube here, not very much at all. How many microbes do you have just in 1 centimeter cubed of soil? Well, give you an idea, we've got some numbers here that I've extracted from the literature. So if you're not familiar with the notation of 10 to the 9 or 10 to the 8, 10 to the 9 just means put 9 zeros after it. 10 to the 7, put 10 zeros after it. 10 to the 10, put 10 zeros after it. So there are colossal numbers of bacteria in soils. And as you can see, if you say go into a forest and a nice, good soil under a leaf litter in, say, a deciduous forest, colossal numbers of bacteria. In more open conditions, in pastures and arable, rather fewer. And what you also see is, again, diversity. And so this is genetically distinct types of prokaryotes, and all these are data from sites in Norway. And the thing to note there is particularly the arable soil has a far lower diversity. So the numbers aren't particularly low, but diversity is. And so one of the things that we seem to be doing with our agricultural soils is reducing biodiversity tremendously. Although some organisms that have been benefited by what we do really, really well. So you can have high total numbers of individuals, but low diversity. And the way to think about this in the bigger animal things is, think cockroach, think rats, things we do to the environment which are bad for an awful lot of organisms are really rather good. If you're a cockroach or a rat, you go, yeah, humans like them. They're producing really the sorts of conditions that we like. And you'd see similar, lower numbers, but similar sorts of patterns if you were looking at, for example, protist eukaryotic microbes. And I'll show you some data for fungi in soils in a little while. So let me focus a little bit on protists, that is eukaryotic microbes with the same sort of cell structure that we have, amoeba and things. So this comes from a paper that a lot of us published. There are actually 49 authors on this paper, and it was an attempt led by Stefan Geisen to gather together the community of people who are interested in protists in soils, all the way from PhD students. Some of the authors were just PhD students, others were Professor Emeritus, retired professors, and everyone in between, to try and identify what we knew, and particularly what were the key questions for the next decade or so when it comes to protist eukaryotic microbes living in soils. And so one of the things you can see is that some sorts of organisms are well studied in soils. So there's a large number of studies on bacteria and quite a lot on fungi, and they have increased during the course of my lifetime. This is quite a nice graph, because at the left-hand end of the graph, I'm an undergraduate, and then it sort of runs, so it runs by accident all the way through my science career pretty much. And one of the things that's happened from about the mid-1990s onwards is increasing numbers of studies on soil bacteria and also on soil fungi, but the protists, which are the brown line there, still really haven't taken off. And yet, as I'll try and show you in a minute, they're really rather important in understanding what's going on in soils and in understanding what's happening particularly with nutrients in soils. And that's clearly rather important when it comes to agriculture. So just from the same paper to show you the sort of diversity we got. So this is a whole range of different sorts of protists, different sorts of eukaryotic microbes that you have living in soils, from really rather small, only a few microns across, all the way up to things that are just about big enough to see with the naked eye. So there's this huge biodiversity of eukaryotic microbes living in soils. And in general, it's the bigger ones that we know a lot about, because the bigger ones have been relatively amenable to study with microscopes. The small ones are a size which are really quite hard to study with normal microscopy. And it's only now that we started using molecular techniques where you take soil samples and you find the organisms by looking for their genetic material in them, that we're starting to be able to study these small things in more detail. So historically, it's been the bigger things that we've actually know more about. But I mean, they are very, very beautiful in their own right. They're absolutely just as wonderful to look at under a microscope as any of the big things that wander across a TV natural history documentary. So I want to introduce briefly why protists in soils, and particularly in agricultural soils, might be so important by showing you this, which is a really rather special field. This is at Rothamstead, which is one of the oldest agricultural research stations in the world, just near London. And this particular field here, which is the Broadbalk, winter wheat experiment has been running as an agricultural experiment since 1843. It's one of the longest running agricultural experiments in the world. They've been growing wheat on this field continuously under various different treatments experimentally since 1843. And the reason why it's particularly important to me is actually it is one of the places where we first started to realize that protists, that protozoa, are actually important in soils. And this was around about 1910. And John Russell, working at Rothamstead with a variety of colleagues, did various experiments on soils from this site where they were interested in how important microbes are for the functioning of soils and allowing you to grow crops. And what Russell did is he took soils into the laboratory and tried sterilizing them to see whether killing off all the microbes then affected how good your crops were when you grew on them. And it's a really nice story in itself of just how science works, because Russell and co messed up some of their experiments and they only partially sterilized them. They didn't heat them as high as they intended. And in his report on these experiments, Russell says, this caused no little annoyance at the time, that it had gone wrong. But rather like Fleming with his contaminated cultures going, penicillin, that's interesting. Russell looked at what he got and went, actually, we may have cocked this up, but it's actually rather interesting. And what he found is that this partial sterilization allowed plants to grow better. And at the time, people thought that if you had protozoans in soils, they were of no ecological interest. Protozoas are water-living organisms. If you find them in a soil, they've just got washed in accidentally, and they're actually doing nothing important there. And Russell went, hang about a minute, this partial sterilization is telling me that they might be, because they're not as robust as bacteria. So it's partial sterilization, he thought it killed the protozoa, but left most of the bacteria okay. And what he thought was, protozoa are eating the bacteria. Bacteria are good for the plants. If I get rid of the protozoa, more bacteria, and my plants grow better. So he had an experiment that had gone wrong that led to a really important conclusion that protozoa are important in soils, but his mechanism turns out to be completely wrong as well. Almost certainly what had happened is by killing stuff, he'd released a lot of nutrients from them, and it was those released nutrients rather than the actual killing of the protozoa that was causing his plants to grow better. Because we now understand that in soils like this, protozoa are very important for the cycling of nutrients. And so let me briefly show you what happens. So within the soils, the protists are feeding on bacteria and other things. They need energy. To get that energy, they tend to actually eat more organisms than they need as far as their nitrogen content is concerned. So they excrete that nitrogen. We do the same. We've got quite a lot of nitrogen. We excrete quite a lot of nitrogen because to get the energy we need to eat often more food than is required for our nitrogen needs. And so they are helping to produce nitrogen and put it into the environment. There are also possibilities that because there are different amounts of carbon to nitrogen in bacteria compared with protozoa, these also makes these things a bit more important. But the feeding of protozoa on bacteria and fungi is helping to release nutrients, particularly nitrogen, into the soils to be used by crops. I also, before finishing, just want to say a little bit about fungi, in particular mycorrhizal fungi in soils. So at the bottom, you can see there a flower agaric. That's the fruiting body of a fungus, the thing that grows to release its spores so they can move to other sites. But most of fungi are what you're seeing at the top there, the mycelium growing through the soil. And indeed, in the case of mycorrhizal fungi, these are fungi, beneficial fungi that can live in plant roots. As many of you probably know, then these mycelia can actually connect individual plants, and potentially material can flow between plants along these mycorrhizae, in a thing that's actually now tends to be referred to as the Wood Wide Web. But mycorrhizae in general are mutualistic. The details vary, and if this was if I was focusing the entire talk on mycorrhizae, I could talk about a lot of details. But broadly, in many situations, the mycorrhizae tend to be beneficial to the plants, and the plants are beneficial to the mycorrhizae. And the majority of land plants have mycorrhizal fungi. So fungi are an important part of the root structure, effectively, of the majority of land plants. And so it's interesting asking questions about, well, what does agriculture do to these things? And here is an early paper from Alistair Fittis' group in York in the 1990s, and they looked at the diversity of mycorrhizal fungi using molecular techniques in both woodland soils and nearby arable soils in Yorkshire. And the details of this complication don't matter, but what I want you to notice is that within the wood, there was a much wider range of mycorrhizal fungi to biodiversity than in the arable. Although again, that thing about humans altering things and doing one or two things doing well. So some things in the arable are doing very, very well. What we've done are suiting them, but in general, we are reducing the diversity. And indeed, actually, this is actually the first scientific paper that used Wood Wide Web in its title. It had been coined only a year before as a cover story on Nature, referring to a different study. And actually, I've talked to Alistair. The reason that they called their study Ploughing Up the Wood Wide Web is because it had been used as a nice catchy phrase on the cover of Nature the year before. They thought if they stuck it in their title, the editors of Nature would like that and be more likely to accept their work for publication. It looks like it worked, and they did accept it. And so this is actually the first use in a title of a scientific paper of Wood Wide Web. But the key thing in this old study here is that in the arable, there's much less diversity of mycorrhizal fungi. Let me briefly show you a modern study. And here we have complicated looking diagrams, which I don't want to spend time on, but we have from a whole collection, it's about 60 different farmers in Switzerland, conventional agriculture, no-till agriculture, and organic agriculture. And you can see, particularly in the organic, vastly more diversity and more complexity in the fungal systems in the soil, and the lowest complexity and diversity in the conventional agriculture. And what we seem to be doing is, many of the functions to do with nutrient cycling are things that would normally be done in the soil by microbes and fungi. Recently, we've tended to say, oh, we don't want to bother with that. We add huge amounts of chemicals and things to do the job another way. The downside, of course, of that is there are all sorts of environmental downsides to that high level of chemicals. You see it, for example, in things running into lakes and running into shallow seas and causing eutrophication and algal blooms in many different ways. And so clearly it would be nice to go back to making more use of the natural microbes in the soil. Now, organic agriculture by itself may not do this because its production levels may be too low for the number of people we've got on the planet. But there's a growing interest in can we mix and match? Can we take some of the bits of modern high input agriculture, but some of the more natural ecological processes of microbes in soil, and put them together into something that's better and something that requires fewer chemicals than we currently use. And the idea I just want to finish with, just to give myself time to look at some questions, is another way that people have been looking, and following on from this, is people have been thinking about nature reserves in slightly different ways. This is a Lancashire Wildlife Trust Reserve, Mere Sands Wood, in a highly intensively farmed bit of Lancashire. The bottom aerial photo there, you can see the reserve marked out in red, surrounded by intensive agriculture. This, you can see that in the top, whereas on the reserve itself, which actually flooded former sand extraction site, there's woodlands, there's heathlands, there's lakes, there's all sorts of things. And one of the things people have been starting to go, well, actually, if microbes are so important to trying to get good nutrient cycling and other things going in soils, and we have greatly reduced the diversity in the sorts of fields you're seeing at the top there, actually, nature reserves are actually a reservoir where many of these microbes may survive and may be able to recolonize agricultural systems if we start to alter the way in which we're running them. And a few years ago, we looked at Mere Sands Wood with particularly a very good master's student called, at the time, she got a PhD now, called Angela Creevy, did most of the hard work on this. And we looked at two groups of microbes living on the reserve, and we found 41 species of testator neebing, 52 species of diatoms just living in the soils. Again, I like this because it illustrates just how little we know about microbes. Before we did this work, the species lists for the reserve had almost no microbes on them at all. All of these testaceous amoebae were new for the reserve, all of the diatoms were new to reserve. Very hard to go onto a nature reserve, anywhere, say in Britain now, and find birds or plants or butterflies that they've missed, but it's dead easy to do it with microbes, because in the majority of sites just haven't been studied much at all. Okay, so what I think that's probably a good time to actually stop and close this, and we'll have a look for any questions.
Well, thanks very much, Dave. I did pick out a few questions, and I'll just, while you can look as well, I have a question from Grace Onions, which says, has any research been done into the effect of electromagnetic radiation on microbes? Before your talk, we had a very interesting one on the potential wide-ranging impacts of the 5G rollout of information technology, and concerns raised about electromagnetic radiation increasing exponentially.
Not really, and I doubt it's any sort of problem. The things that we've been doing by changing land use, by adding huge amounts of nutrients, and also by changing the climate, are just so significant that anything, any minor effect there may or may not be, and I suspect there probably isn't. And I mean, soil will actually screen you against those sorts of things very easily. So no, I don't think it's an issue.
Well, Kevin Webb asked another question, and this is, will we find microbes in the solar system? Now, I think there's already been some exploration of this, but can you go into that a little more in an answer?
I can, yeah. So, I mean, one of the things that's interesting, I was talking about Tommy Gould's ideas of the deep hot biosphere, and the fact that we now know that deep within the Earth, there are lots of microbes living in the rocks, and this can be a kilometre or so down. If you imagine smashing a large asteroid or something into the Earth, you will spin off all sorts of bits of rock, and hidden within those rocks there will be microbes, and they can potentially land on another planet, and some of those microbes may survive. So and it's actually because the Earth is bigger than Mars. In the past, if things did this, they probably went the other way, because the Earth is bigger, it has a stronger gravity and attracts rocks better. And so one possibility is that actually life may have arrived on Earth from Mars when Mars was wetter and had life earlier on. So it's possible that if we were to find, say, a little bit of remains of life on Mars, which is probably unlikely but not absolutely impossible, it may turn out to be really boring. It may just turn out to be, related to us, admittedly a very long time ago, rather than having evolved separately. Yes, so I mean, microbes can potentially move around potentially like that. So I mean, there's this big question, what would be really nice to find is life that is a separate evolution of life, and to find out, well, what does that look like? But it's quite possible, at least within our solar system, if we did find more microbial life, it may be effectively our ancestors, they're on the same family tree as us. Admittedly, we split a very, very long time ago.
Okay, an interesting question from Holly here, who asks you, does vertically farmed farming does the food lack in nutrients?
What is meant by vertically farming in this?
Well, I just picked it off. She's just put this question on. It's towards at the end. Do you agree vertically farmed food is lacking in nutrients?
I presume that's when you actually build.
So these are hydroponic systems, are they?
Yeah, it's building.
Holly's just saying grown in water. Yes, so these are hydroponic systems. So again, what you're doing there is effectively what we have been doing with intensive agriculture anyway. You're saying we don't need to bother about the microbial processes for nutrient cycling things, because we're just adding all the nutrients ourselves. You can actually use natural sources. So I mean, if you're growing stuff in, say, ponds which have got the nutrients coming in from manure and things, has been done in, used to be done in parts of China and things, then those are being processed by microbes. They put in a hydroponic system, we're effectively adding the chemicals. So we're sidestepping the need for all the microbes. And this is largely what we've done with modern agriculture, high input agriculture. We've made the importance of microbes in nutrient cycling and the other roles that they may take, for example, in controlling pests and things, unimportant, because we've put in all the nutrients and chemicals. We've done the pest control with chemicals. And this works. The problem is, of course, it has a lot of environmental downsides. And so it looks to me and quite a lot of people that the interesting way is to try and find, going forward, is to try and find something that reduces chemical use by understanding more what the microbes do and making better use of those, and therefore allowing us to actually use fewer chemicals. And I think these chemicals anyway cost money. So you're potentially saving the farmer money if you can do it as well. I mean, the ultimate one is to just do everything totally organic, but that looks a bit of a long shot because there's just too many of us to feed, and the production from totally organic systems is probably not good enough. So some sort of best of both worlds, if we can construct it, looks to be the way forward to trying to reduce the colossal environmental impacts of high input agriculture, but still producing the food that we actually need to feed people.
Yeah, I was very interested in your point about that Swiss study that looked at the nutrients, the multiple nutrients in organic farming and no-till farming, and then the current intense agricultural industry we have now. I mean, is there a balance? Were you saying that intense agricultural farming still has a place, a good, a reasonable place in the whole system?
There are multiple ways of doing that. My suspicion is the answer is yes in certain situations. And you often in environmental discussions, you end up, people arguing, oh, we should do everything like this. My suspicion is that, there is undoubtedly a role for organic agriculture. There is, I think, a role in some places for limited high input agriculture. But if we do most of it with this high input agriculture, the amount of environmental damage we're doing is really rather great. And so clearly we have to produce enough food to feed ourselves, but if we can be more intelligent with microbial ecology of soils and make better use of these more natural processes, and therefore reduce the numbers of chemicals that we're using, this is, from an environmental point of view, good, and also potentially economically good, because all of these chemicals cost money.
Yeah, is this research still something very much in progress?
Yeah, so and include, when you're talking, as you saw from that graph, we know most about bacteria. Increasingly, fungi are being studied. Really, the protists and the role of protists has just been bubbling along as not particularly well studied at all. And one of the reasons for that paper that I was talking about, where we had sort of nearly 50 of us gathered together to try and set out an agenda for the next decade or so with protists, to work out really what we need to know. And the really interesting thing is that it's actually, this is an area in which it's now possible to make progress with modern molecular techniques using DNA and RNA within environmental samples. If you think about trying to do stuff in soils, which I've done quite a bit over my career, with a microscope, it's horribly difficult to find any microbes in soil because you can't see through the soil, and you end up trying to dilute a soil sample under your microscope side and hunting between all the soil particles looking for the microbes. It's slow, it's difficult, and you're very limited what you can do. So that, the changes in technology that's been over the last decade or two has completely opened up what we can do with microbes. I would, regularly do argue with, stood before, a group of particularly younger researchers in ecology and try and go, look, microbial ecology is now where it's at because it's desperately important. And up to now, it's been really hard to do. Now we have the technology to actually make, leaps and bounds in this sort of area. And, I think this is an area which will change tremendously. And probably most things I say will be out of date quite quickly over the next, 1 to 2 decades.
Yeah, I had a question, which is, you mentioned earlier about the increase in harmful microbes in shallow water areas and in rivers. caused by heating and intense agricultural runoff. And I think there's been some research done on this about how we can perhaps harvest this runoff to actually create commercially viable energy. Is there anything you can say about that?
Yes, people do. I mean, you can grow biomass. So I mean, if there are particularly tropical systems where various quick growing plants are being grown in ponds which have got this runoff coming into them, and then you can then harvest that as energy, and at the same time you are removing some of this runoff. And the way it's often been done in more temperate places like Britain is things like reed beds, and using reed beds for processing wastewater and cleaning it up. And it works, but you need quite large areas of reed beds. And again, actually one of my colleagues, Jane Fisher at Centre for Alternative Technology, to work on this some years ago. It is the microbes in the reed beds that are often as important as the reeds. You often need fairly extensive systems to process the water that you're flushing it through.
Yeah, and I think we've just got time for one more question. I'm just reading here from Luke Crosby, and he's saying, can you read it at the end? It says, what can microbes teach us about how ecosystems might respond to short-term ecological changes as a consequence of climate change? And can they be used to make predictions on future outcomes? It was the last question.
It's a really good question. The answer will be about an hour long. So here's the very quick. So one of the nice things about microbes is they have very short generation times. So you can actually do experiments on the microbial ecosystem on timescale of days to weeks, that if you were doing it with plants and animals would take you years. There are obviously then questions whether you can scale up or not. And indeed, people have attempted to do this. So, and have attempted to construct little systems of microbes and or very small plants in sealed containers where you can actually look at global change and look at changes in CO2 levels and changes in temperature, and to what extent the ecosystem can bring down CO2 levels. They're very difficult to do, and they've been very difficult to fund. There are enough studies to make these look quite interesting, and they do sometimes give slightly more optimistic results than, for example, the big computer models of climate change and the effects of life. The difficulty is they are very expensive, very difficult to do, and hard to fund, partly because people say, well, we can do this on computer models as well. Actually, it's probably very useful to have multiple approaches. So they're good as experimental systems, because you can construct an ecosystem, a whole ecosystem that you can play with on a timescale that doesn't take your entire career. So it's effectively a different way of approaching questions which otherwise you would approach in a big computer model. They can be really, really interesting. And clearly microbial systems can respond to change quite quickly. So over the course of 1 year, you get huge changes, for example, in a lake as temperature changes and as nutrients change, in a way that, if you're studying trees, it would be 3 or 4 generations of people required to do the study.
Well, thanks very much indeed, Dave. A very interesting talk. And